Okay, chapter 3 will be on Mendelian genetics, and this is another mostly review chapter, so hopefully you've encountered most of this material before. I'll go through things in some decent amount of detail, just in case you haven't, but hopefully most of you can kind of breeze through this chapter. So these are kind of the goals, the learning goals of this chapter, defining and distinguishing between phenotypes, characters, traits, and phenotypes. So what exactly do we mean by those three different terms? And then DNA-based information, gene, allele, and genotype. What do those terms mean? I want you to be able to determine the phenotypes and genotypes of individuals by analyzing the phenotypes and or genotypes of their ancestors or offspring. So this is kind of the basics of genetic analysis, is you do a cross between individuals. Some of them you know their genotype, some of them you don't, and then you use basic logic in order to figure out the missing pieces. Understand the difference between a natural and artificial population. Describe how geneticists use artificial populations for genetic analysis. Integrate probability rules to the analysis of simple genetic traits. How does stochasticness play a role in these crosses? Apply a chi-square test to genetic crosses, so determine if the experimental data you see matches your expectations, and then differentiate the genetic basis of traits and genotypes of individuals by analyzing pedigrees. So pedigrees are what are used in order to typically analyze human phenotypes, and typically this means human disease. So understanding the genetic basis of human disease, how do you do that using pedigrees? So the father of genetics, Gregor Mendel, so he was a monk, and prior to his work, many believed in blended inheritance. And this is the theory that offspring receive a mixture of characteristics from their parents, resulting in offspring with intermediate phenotypes. And so, some of this makes sense. Some of this, should make sense. The reason they thought this is because there's a lot of things about the world that kind of fit this model. So if you have a very tall father and a very short mother, typically that means that you're going to be kind of in between. But he kind of turned this whole idea on the head, and he proposed this particulate inheritance and the theory that characters are determined by discrete units that are inherited intact through generations. And so nowadays we have a very good molecular basis for this idea. At the time there wasn't a lot of knowledge about the cell that would help us make sense, but now that we know a lot about the characteristics of DNA, this is kind of very straightforward now for us to think about. And so he studied this again before we knew what the cells were made out of, the molecular characteristics of the cell, and he made many observations through the study of pea plants, and as a result he proposed a number of rules. So these are rules for the pea plants that he studied. So we are going to extend some of these rules in future chapters, but for now we're talking about the plants that he studied. So characters are unitary, so that is they are discrete. So typically there's only two different types of traits that a character can have. So flower color was either purple or white. Height of the pea plant was either tall versus dwarf. Genetic characteristics have alternate forms, so he defined genes, and each one of these were inherited from one or two parents. And so these alternate forms of the genes are called alleles. So that what we call them now would be called alleles. One allele is always dominant over the other, and the phenotype reflects whatever the dominant allele is. And so again, this is just true for the pea plants that he studied. This is also true for many traits today, but we're going to explore in the next chapter when alleles are not dominant over each other. So this is just the rules that he followed. Gametes are created by random segregation. Heterozygous individuals produce gametes with an equal frequency of the two alleles. And then different traits have independent assortment. And so in modern terms, genes are unlinked. And typically what this means is that they are on different chromosomes. So we now know there's many exceptions to these rules, so we're going to cover these in chapters 4 and chapters 5. But for now, just for this chapter, if I tell you I'm asking a question based upon chapter 3, you should assume that these different rules are hold true. So there's going to be 4 parts of this chapter that we're going to cover. So we're going to cover the analysis of single character in artificial populations. So this is the work that he did on pea plants. The second part is we're going to do analysis of single character in natural populations. So this is how we apply the findings of Mendel to the study of human population and human diseases. Part 3 will move on to the analysis of multiple characters at the same time. So if we look at the height of the pea plant and the color of the flower of the pea plant, what happens if we have mixtures of both of those characters? And then finally in part 4 we'll cover chi-square tests.